Hi guys, so here you are in this video watching how I am going to use this PhET simulation, which actually helps you visualize how a lac operon in an E. coli functions. So this simulation will show you two structural genes and a regulatory gene storage room. So I'm going to take out all these genes and place them. Ideally, this is the DNA of the E. coli bacteria, and in a real life scenario, this DNA is circular. Okay, so you have to imagine that the ends are connected, and it's a circular DNA. So as I am going to work around my way through this simulation, I will ask you to pause and answer the questions asked in the worksheet. So you need to predict or observe what I am doing on this simulation and write your responses. Okay, so let's go ahead and begin. So the first thing that I'm asked to do in the worksheet is position the lac promoter on DNA and then click on the legend button. So this is my lac promoter and I'm going to place it. You can see a yellow prompt is showing, so I have to place my promoter over there and observe what happens when I place the promoter. So I will now switch on the legend so you can know what that blue structure is which is floating. You now pause the video and describe what you saw. What happened when I placed the promoter and clicked on the legend? What did you notice? Thank you for pausing. Next, I am now going to position the LAC-Z gene on the DNA. As I position it, you will pause the video and describe what is happening. The LAC-Z gene is on the DNA. Observe what is happening. And you can now pause and write your response to the second question. Thank you for pausing. On the left hand side top corner, you must have noticed a lactose injector. I can manually inject lactose or automatically. So in question number 3, I am asked to use a manual lactose injector to inject some lactose in this cell. So this place represents a bacterial cell, and I'm going to manually inject some bacteria in small quantity. Uh, Bacteria actually absorbs these lactose from the agar medium in which they are being cultured. So you have to imagine that you are culturing this bacteria in an agar jelly, and in that agar jelly there is a lot of lactose. Now I'm slowly making the bacteria absorb small quantities of lactose. Look at how the lactose is getting injected manually. As I am clicking, lactose is entering the bacterial cell. Observe what is happening and explain the role of the product of LAC-Z gene. What is the product of LAC-Z gene? First identify that and then notice what is the LAC-Z gene's product doing to the lactose, and then explain it in your answer in relation to bacterial respiration. You can now pause the video and explain your observation. Thank you for pausing. I'll now move to question number 4. With only the LAC promoter and the LAC-Z gene in position on DNA, beta galactosidase enzyme is made all the time even if the cell is not taking up any lactose. Suggest why this would be a disadvantage to the bacterial cell. So you're noticing that on the DNA right now, there is a promoter for the LAC-Z gene and the LAC-Z gene. The RNA polymerase is actually reading the LAC-Z gene and transcribing its mRNA, which is later getting translated to the beta-galactosidase enzyme. So the question is asking, when there is no promoter over here, the LAC-Z gene is constantly getting transcribed. How is that a disadvantage to the bacteria? Even when I have not injected any lactose, the gene is getting transcribed. Pause the video and think about what disadvantage the bacteria is facing right now and answer it. Thank you for pausing. I hope you entered your response. Question number 5. Introduce LAC operator. Now I'm going to introduce LAC operator. I'm going to introduce the regulatory LAC-I gene and the LAC promoter. So all these gaps are going to be filled. Let's see what happens. So I'm introducing LAC operator. I will introduce LAC-I's promoter. Then I'll introduce LAC-I gene. And we will observe what is happening here now with no lactose. Observe what is happening. You can see an RNA polymerase recognizing the LAC-I promoter. After reading the promoter, it goes ahead and starts transcribing the LAC-I gene. So note what is being produced. And then note what happens to that black thread. Keep looking at the legend to identify what each structure is. So you saw LAC-I repressor actually going and binding to the promoter. Continue observing what happens now. So we can see the repressor binding the promoter. So what? What next? Okay, did you observe what happened now? Now pause the video and record your response to what happened when the repressor bound to the promoter. Okay, thank you for pausing. Now I will move to situation asked in question number 6. Add some lactose. How does this affect transcription of LAC-Z gene? Let's add some lactose. And let's see how that will affect LAC-Z gene transcription. You can see lactose floating in the cell cytoplasm. And quickly they catch up with the repressor. So repressor is now busy with lactose and has left the promoter binding site. Observe what the RNA polymerase is doing. Okay. Now you can pause the video and answer question number 6. Thank you for pausing. I will now move to question number 7, situation. So you observed that the LAC-Z gene is now again getting transcribed and it's producing beta-galactosidase enzyme. What is the beta-galactosidase enzyme doing? It is hydrolyzing the lactose. And now what is happening is you have to write how the LAC-Z gene is getting switched off. Okay. Note all the lactose have been hydrolyzed by the beta-galactosidase enzyme. So what is now leading to blocking of this LAC-Z transcription? Pause the video, write your answer. Thank you for pausing. I will now move to the last question. So in the last question, I'm going to move to the next tab. You can see right now we were watching about lactose regulation. In the next part, I'm going to move to lactose transport. In this tab, I am showing you the cell membrane of the E. coli bacteria, and I am going to place all the relevant promoters and operators and genes in their position. These are the RNA polymerases floating in the cell cytoplasm. Your job is, after I place everything on this DNA, that means after all the genes are in their correct order, describe what happens when lactose is added and explain the role of the protein product of LAC-Y gene. So after I place all of them, LAC-I promoter, LAC-I gene, LAC-Z gene, LAC-Y gene, LAC promoter, and LAC operator. Everything is in their place. I will now start injecting, not injecting exactly, I'll show the legend and observe what is happening on screen. You're noticing all the genes are now getting transcribed and some mRNA forming, and that mRNA is forming some enzyme. Make sure you're using the correct terms for these formation. And there are some new proteins that are being synthesized which are going to the membrane. Now I'm going to manually release some lactose in the agar jelly and see what's happening. Observe. Think about what did LAC-Y gene produce? What was transcribed and what was translated from LAC-Y gene? And what is that product doing? The LAC-Y gene product. Hope you're noticing. Let me quickly increase the speed of all of these motion so that we can see what's going on a little faster. We can see lactose entering through that green color protein into the cell. Make sure you are using the correct terms as to what is happening to the lactose because of LAC-Y gene product. And whenever you are ready, you can pause the video and describe what happened when all of these genes were in their place and the lactose was there in the agar jelly. Describe what happens in this scenario in answer number 8. Thank you for pausing. You will now move on to check your answers and compare your answers with the answers shared in the mark scheme, and let me know if you have any questions in the reply to this assignment. Thank you.